This is a story of the positive feedback loop involving insulin and osteocalcin, and how a hormone from bone can help regulate insulin secretion, but also synthesis within the pancreatic beta cells. So insulin, following a meal, will be produced and released by the pancreatic beta cells, typically in response to glucose, but also branched chain amino acids like leucine. And if insulin's in circulation, it can bind to insulin receptors that are present on cells that are insulin dependent. And some of those cells include muscle cells, which have insulin receptors, or adipose or fat cells, which have insulin receptors, but also bone cells will tend to have insulin receptors, specifically osteoblasts. So this is a depiction of an osteoblast. Osteoblasts tend to deposit bone. And right here, you can see this is our little insulin receptor right here. And insulin will come over and it'll bind to the insulin receptor. It'll activate the two alpha subgroups of the insulin receptor, which trigger a conformational change in the little beta subgroups right here. And the beta subgroups will then autophosphorylate themselves. So what they do is they take ATP and they transfer the third phosphate from ATP onto themselves to form ADP. And what happens is when those two beta groups are phosphorylated, they recruit something called IRS1 and 2, or insulin response substrate 1 and 2. And once these two guys are recruited and activated by those beta subunits of the insulin receptor, they'll go over and they'll turn on a protein called PI3K, which stands for phosphoinositide 3 kinase. And what this does is it's a kinase, so it's facilitating the transfer of a phosphate from something onto something else. In this case, it's transferring a phosphate from ATP onto PIP2 to form PIP3. So what it's doing is it's transferring the third phosphate on ATP, the gamma phosphate, onto this molecule on the third hydroxyl group to form this molecule right here. And once I start forming more and more of this molecule, it'll recruit this protein called AKT, or protein kinase B, so another kinase in the story, and it'll kind of recruit it towards the membrane and turn it on. And what this protein does, AKT, is it's a kinase, so it also facilitates the phosphorylation or the transfer of a phosphate from one thing to another thing. But what it does specifically is it sticks a phosphate on this protein called FOXO1. So it sticks a phosphate right on here on FOXO1. And FOXO1 is a transcription factor, which is involved in regulating a lot of genes, but when it's phosphorylated, it can no longer act as a transcription factor. So if it's phosphorylated, like it is in this case, it can't move into the nucleus and then bind to enhancer regions of genes and facilitate the transcription of those genes and the eventual translation of those genes into proteins. So when it's phosphorylated, this FOXO1 gene is deactivated. But normally FOXO1, I said gene, the protein, but normally FOXO1 is a protein that goes and turns on genes associated with producing this protein called osteoprotegerin, or OPG. And so this protein right here is produced when FOXO1 is available within the cytoplasm in its unphosphorylated form, which allows it to move into the nucleus. But if insulin's around, if insulin's binding to the insulin receptor, FOXO1 is phosphorylated, therefore we are producing less of this protein. And this protein, what it does is it's released out into the interstitial space of this osteoblast, and it kind of looks like a little receptor, kind of like this. It looks like a little receptor. It's produced and released out into the interstitial space. And what it does is it mimics this receptor right here. So it mimics the rank receptor. And the rank receptor is sensitive to a molecule called rank ligand, which is produced by the osteoblast and osteocytes in response to both hormonal environments and mechanical stress. So if I take off running, or I have a hormonal environment that's kind of inducing me to remodel my bones and change my bones, what happens is these cells will produce rank ligand, which will typically go over and it'll bind to the rank receptor and trigger downstream effects in this cell called an osteoclast. So this is our osteoclast, which is facilitating bone remodeling, or the breakdown of bone and then the eventual resynthesis of that bone by the osteoblast. But what happens is this rank ligand right here, it typically can come over and bind to this rank receptor and then activate downstream pathways which facilitate bone remodeling. But if this protein right here, the OPG, is produced and released, it mimics the rank receptor and acts as a decoy receptor. So this rank ligand will come over and bind to this instead of binding to this. And so the issue is the more of this that is present in the interstitial space, the less the rank ligand is actually binding to the rank receptor. But what happens when insulin is around, if insulin is present and insulin binds up here, I phosphorylate FOXO1, so I'm decreasing the synthesis of this protein. Therefore, the decoy receptor is no longer around, which allows the rank ligand to come over and bind to the rank receptor, which will activate this rank receptor. And the eventual downstream effect of this rank receptor is I'm causing the extracellular matrix of bone to become more acidic. And we'll talk a little bit about how that happens. But if this extracellular matrix of bone becomes more acidic, it starts breaking down the bone, breaking down the hydroxyapatite and the mineralization of that bone, so that I can remodel the bone to better adapt to whatever the stress or the strain was that I experienced during exercise. And so this happens by the rank ligand binding to the rank receptor, which then trimerizes, so it comes together, 3 proteins come together. And what they do is they activate this protein right here called TRAF6, or the tumor necrotic factor receptor associated factor 6, which then goes and it activates a bunch of these downstream signaling pathways. And I just kind of included this for completion, but these downstream signaling pathways eventually activate this transcription factor, which regulates genes. And some of those genes are then transcribed and translated into functional proteins, which include carbonic anhydrase and this vacuolar ATPase. And so this carbonic anhydrase right here, carbonic anhydrase, what it does is it facilitates a normal reaction that happens all the time, where water and CO2 react together to form bicarbonate and hydrogen ions. And this is carbonic acid. It's just an intermediate that's formed, but eventually that hydrogen ion just breaks off and we produce a free hydrogen ion or proton and bicarbonate. And then what the cell does is it also facilitates the transcription of this guy right here, the vacuolar ATPase, or the translation of that guy to a functional protein, which allows me to pump this hydrogen ion out into this space. And so I'm breaking down bone now. I'm breaking down bone because I'm making it more acidic. I'm decreasing the pH of the extracellular matrix. But what also happens is I'm starting to decarboxylate osteocalcin. So typically osteocalcin kind of looks like this, where it has glutamate residues. So right here, this is my glutamate residue right here. This is my glutamate residue. So it's an amino acid, but what happens is because of the enzyme that's vitamin K dependent in the osteoblast, I've added another carboxyl group to this. So I have 2 carboxyl groups that both have negative charges right here and here. And those 2 negative charges coordinate with the +2 charge of calcium, making it really bind tightly with hydroxyapatite. But what happens is when I make this extracellular matrix more acidic, or I decrease the pH, I can come over here and those hydrogen ions can facilitate some organic chemistry where I'm decarboxylating this. So I'm decarboxylating this. I'm eventually losing CO2. So I'm getting rid of this little carboxyl group right here in the form of CO2. And so now this osteocalcin molecule right here is decarboxylated. It's no longer really binding to calcium as tightly, so it's able to kind of bounce off and move into circulation. So during exercise, and then following that exercise in the presence of insulin, when I'm remodeling bone, I'm also producing this molecule and releasing this molecule into circulation. And this is having an effect on the system-wide metabolism. So it's having an effect on muscle cells, on fat cells, on cells in the brain. It's having an effect on pancreatic beta cells. And what it's doing is it's facilitating an adaptation to exercise. And so this little molecule right here is osteocalcin molecule that was decarboxylated due to the decrease in pH caused by the presence of insulin and all this little pathway right here. This molecule can come over and bind to a G protein linked receptor on a pancreatic beta cell. And if it binds to this G protein link receptor, a type C G protein link receptor, it can cause a bunch of downstream effects that I'm not going to go into in this video, which facilitate an increase in gene transcription of insulin. And then I'm increasing the rate. I'm converting pro-insulin into proinsulin, and then proinsulin into insulin. So I'm facilitating the post-translational modification of insulin into the active form, which can then be released in the presence of nutrients. And then I'm increasing the rate of secretion of insulin. So overall, this molecule, this osteocalcin molecule, which is released in response to insulin and mechanical stress during exercise, is facilitating the ability of these pancreatic beta cells to produce insulin. So I'm enhancing their ability to produce and release insulin in response to a meal. And then that insulin can then feed back. It can feed back and further enhance my ability to produce osteocalcin, which further enhances my ability to produce insulin. So overall, this is a mechanism that's definitely left out some details, but it's all about how bone and the pancreatic beta cells participate in a feed-forward reaction or a coacting response where they're monitoring body-wide energy levels, integrating those body-wide energy levels with exercise, producing this molecule insulin, and then insulin's regulating the production of osteocalcin indirectly, but overall, it's a feed-forward response that facilitates the metabolic health of an organism.